Hello, my name is Boston Simmons. I'm a research assistant in the lab with Dr. Liza Mikowski. I'd like to thank the coordinators for inviting me to this event, and I'd like to present my work on novel preclinical recombinant inbred model to identify genetic modifiers of breast cancer. So, breast cancer is a disease that affects 1 in 8 women in the United States. It has a variety of risk factors, including age, family history, and different environmental factors. Um, so variability and susceptibility. Not everyone who experiences risk factors develops cancer due to genetics in their environment. This is important to us because our lab studies gene variants that can impact breast cancer susceptibility and its aggressiveness. So, an introduction to unique preclinical mouse model. This is our BXD recombinant inbred family. It's created by crossing the C57 black 6 mice with the DBA2J mice. Um, with siblings matings, each BXD strain represents a unique mosaic of B6 and D2 alleles homozygous at every loci, and it's important to note that this is isogenic. So some of the main cancer models out there right now are the syngenic model of cancer, where you have cancer cells that are injected or transplanted, or you have patient-derived xenograft or PDX, where you have patient-derived cells that are injected into immunocompromised mice. The model I'm going to show today is a little bit different. It's a spontaneous genetic engineered mouse model, or GEM. So the first gem I'm going to show is the C31T antigen or C3 tag mice. So these are FVB mice that are transfected with the SV40T antigen. Uh, the way that this works is that this T antigen blocks the tumor suppressor P53 and RB, which leads to tumorigenesis. In female mice, they develop breast cancer in around 4 to 5 months. And in male mice, these mice develop prostate cancer starting around 9 months. Um, so this is a spontaneous genetic model of cancer where the SV40, how it acts is it blocks P53. This is important because P53 is a tumor suppressor gene that normally works to repair DNA and block cancer. So when there's DNA damage, P53 is activated, repairs that damage, and the cell is either repaired or apoptosis occurs. Therefore, there's no proliferation of these pro-cancer cells. So when you don't have P53, this leads to failed DNA repair and cancer forms. So when the T antigen is present, P53 is no longer able to repair this DNA damage. So the DNA repair pathways are no longer activated, and apoptosis does not occur. This leads to proliferation of these cancer cells and then to tumorigenesis. So this model is a good model because it mimics the slow development of breast cancer seen in humans to loss of P53, but it does not reflect the human genetic diversity that we see. So it does have some downsides to it. Um, so this is the model that we've been working on now. It's a C3 tag cross of a BXD model. So we have our C3 tag fathers, which have the T antigen, crossed with our BXD females. This gets us our F1 progeny of our BXD BC mice. And again, because of the T antigen, we see this breast cancer in the female mice starting around 4 to 5 months. Um, just the way that this cancer model works, you start with a normal mammary duct. This goes to atypical ductal hyperplasia. This goes to then intraductal carcinoma, and then finally goes to invasive ductal carcinoma. A little bit about some of the data we collect in this study. The first thing we collect is latency. This is the time between when the mouse is born to when the first tumor occurs. Uh, then at endpoint, which is determined by our IACUC endpoint, we collect information on tumor burden, tumor multiplicity, and tumor volume. The final thing that we collect is survival. This is the total time the mouse is alive from birth to endpoint. This data is then collected and then entered into Gene Network, and this is some of the data we already have. Uh, So some of the phenotypic variability that we're seeing in the BXTBC strains is shown here. One of the traits that we're showing is multiplicity. And if you look here, we have anywhere from one tumor on the low end all the way up to 15 tumors on the high end, and also tumor burden. And this is calculated by the total tumor volume mass in the mouse. And if you look here, we have less than 1 gram all the way up to 5 grams. If you look at the FVBN or C3 tag parent strains, you can see that it's on the lower end for multiplicity, but on the higher end for burden. This indicates mouse has less tumors, but the tumors on average are larger. A little bit more on the phenotypic variation. If we look here on progression, we can see that when we're looking at both endpoint and latency, that it just has this varies between the mouse strains, as well as how long each mouse is tumor free for. If you look here again at the FEB strain, you can see now with these two traits, it's about in the middle. So in conclusion, we have generated a novel preclinical model of breast cancer and prostate cancer. So this is a genetically engineered mouse model, or GEM. Spontaneous tumor formation is due to the large T antigen. So there's no pregnancy or hormone induction needed. It's immunocompetent, meaning that these are not immunocompromised mice. So you can do different immune assays and you can look at immune function and how it changes in these mice. They're 100% penetrant of the transgene. The female mice develop breast cancer starting around 4 to 5 months, while the males develop prostate cancer starting around 9 months. And finally, the BXT intercross shows heritable variability. I'd like to thank the coordinators. I'd like to thank the lab of Dr. Liza Mikowski, and I'd like to thank our collaborators. And I will now open the floor up to any questions.